So it's our pleasure to welcome today Xiang Tao Ruan, who's going to be telling us about working with massive data sets in the petabyte scale and how he's been able to do that in his postdoctoral work in the Department of Molecular and Cell Biology at UC Berkeley. So thank you for being with us. The presentation will be about 40-45 minutes, and then we'll have some time for questions at the end. So feel free to put your questions in the chat or raise your hand and ask them when the time comes. So I'll pass it over to you.
Yeah, okay, yeah. Thanks very much for the introduction. And also it's a very great pleasure for me to present my research here. So the first question, because today we are here to learn about lattice microscope. So the first question is, why do we need a lattice microscope? To answer this question, let me first compare several commonly used imaging modalities in molecular and cell biology. Let's use the imaging of this cell as an example. So first is TURF, which is based on the total internal refraction, and this can achieve very high signal to noise ratio. However, it can only image a very thin layer above the curved slit. And the next is confocal, which have a larger depth focus. And even though being blocked by some pinholes, this technique have very strong autofocus light that will bleach the unimaged regions. And also, this is a point scan process, so it's the image speed is usually very slow. And the spinning disk is a parallel version of confocal. This can make it much faster, but it still have strong autofocus light that will bleach unimaged regions quickly. Then light sheet microscope is an emerging imaging technique that usually have two objectives that are perpendicular to each other. One is for the excitation and the other is for the detection of the emission light. This particular layout allows for better optical sectioning that can enable better signal to noise ratio and also the higher axial resolution. And the traditional light sheet microscope using a Gaussian-shaped light sheet, and this light sheet still have some strong autofocus light that will bleach the sample. Also, usually this light sheet is typically very thin, very thick, and usually this can only achieve cellular resolution and can hardly resolve subcellular organizations. And the next light sheet, invented by Bazivik Lab in 2014, overcomes this disadvantage by emulating the cell with a very thin light sheet. So it can still have very large depth focus, but it's have much fewer photo bleaching and photo damaging, and also can achieve a much higher axial resolution that can resolve subcellular organizations. So this allows light sheet be very useful for the imaging of volumetric imaging of subcellular dynamics for a long time without worrying about bleaching the sample. So here's a comparison of the spinning disks with lattice sheet microscope for the imaging of endocytosis. That is how cell is. So the image starts from the similar signal to noise ratio, and we can see spinning disk has a 20 to 30 times faster bleaching rate than the lattice sheet. This means we can image using lattice sheet to image the sample for much longer time. So the original light sheet microscope has most been used for imaging a single cell or small regions. However, for modern biology, the imaging of multicellular systems and monitoring their long-term dynamics is becoming more and more important because the imaging of single cells is harder to understand the multicellular natures of the tissues, organs, and even the whole body. So to enable more efficient imaging of the large multicellular systems, we developed the next generation lattice microscope. We call it Mosaic because it contains more learn 10 imaging modalities within one microscope. And all these modalities are equipped with adaptive optics. And compared to the previous version, it enables a much larger field of view of imaging with more than 50 times faster image speed that can reach about 4 terabytes per hour. Also, because we have multiple imaging modes, we can do correlative imaging with multiple modes involved for the same field of view. So in our lab, we have built 2 mosaic systems, and we build the system such that these different modalities can be automatically switched using the control software. So now let's demonstrate how this new microscope can enable us to do new biology with these striking features. So first, mosaic enables the imaging of very fast dynamics of subcellular layers. Here is an example of the imaging of the ER fast dynamics. And here we have a 3 seconds per frame and with very high axial and lateral resolutions. This allows us to visualize ER and ER sheets and how they move in the high spatial temporal resolution. And also, mosaic enables the imaging of a very large field of view that contains many cells. So here's an example of the imaging of oblique illumination phase imaging of 2D imaging of the HeLa cells. And here we have a frame rate of 2 seconds per frame with about 1,000 time points. So this is a millimeter scale field of view. This allows us to monitor more than thousands of cells in the same field of view and monitor their fast dynamics. And also, mosaic enables the live imaging of large field view with 3D volumetric imaging with Lattice Lightsheet. So here is an example for the imaging of the kidney epithelial cells for the nuclear and the ER. And we imaged at image rate at 1.5 minutes for about 1,000 time points. So this is the duration of about 24 hours. This is a really long-term imaging that can hardly be achieved with confocal microscope. Mosaic, so with this large field of view, we can inspect hundreds of thousand cells and to screen some new biology. For example, we can use machine learning models to segment all the nuclei for this large field view, and then quantify some features like we can count how their number changes over time, or we can quantify their volumes by putting the nuclei for all time points together. And since we can seamlessly switch between different modalities, we can also using imaging same field view with multiple imaging modes. So here's an example of the correlative imaging of the same cell with both phase and the lattice lightsheet. So we can look at the overall feature dynamics with the phase and also track the mitochondrial dynamics using the lattice lightsheet. And this mosaic also equipped with adaptive optics, so that enables better imaging qualities. So let me first introduce a bit of background about adaptive optics. Adaptive optics is usually very useful for imaging non-transparent multicellular systems like zebrafish or deep brain imaging. In these situations, the light will get aberrated when going through the sample due to the mismatch of refraction index, which will significantly reduce the imaging quality and make the image looks blur. To solve this problem, we borrow the idea from astronomy that we will have some guide star as a reference. So use the guide star as a reference, we can estimate the aberration of the light, and then with some kind of sensor, and then we can use our feedback units to correct these aberrations to make the light wavefront better, and this will significantly increase the image quality. So here is an example of the live imaging of the mouse brain comparing no AO and AO. So after AO correction, we can see the imaging become much sharper, and it's much easier for us to see the fine details in the imaging, like the axons and dendrites and dendritic spans, even deep in the mouse brain. Yeah, so this being said, the mosaic enables the imaging of large multicellular systems for both live imaging and also large fixed samples. This allows us to monitor the cellular dynamics in their native states with the live imaging, and also enable us to analyze a lot of cells within the large samples with the fixed samples. So now we have a basic understanding of how mosaics enable us to do use new ways of doing biology with its striking features like the fast image speed, large field of view imaging, low photo bleaching, low photo damaging, and also with correlative imaging and adaptive optics. However, this powerful imaging capability brings new problems for the data processing, because even in a single experiment, it may generate hundreds of gigabytes to petabyte scale image data. So here's a list of typical data sets we may get for the either 5D imaging or the super resolution 4D imaging. I need to emphasize that the data sets increase drastically with the complexity of the organism. So for the imaging of mouse hippocampus, mouse brain, and human brain, it may generate thousands, millions, or billions large data than the imaging of the yeast. So we now just reached the capability to imaging the mouse hippocampus. For the imaging of entire mouse brain or human brain, these are our future pursuits. And this large data sets cause a lot of problems for the current image processing tools, that some may be too slow, that requires days, weeks, or months to get final results, and some others maybe fail right away because hardware limits. To solve this problem, we develop an efficient and scalable software, Petakit 5D, to enable petabyte scale imaging processing. So here is the basic workflows for our imaging and also processing. So first, we will store the images into our fast data server with a compressed form, and then we will use our computing cluster with CPU and GPU nodes to process the data. So first, let me go over the features in this software. So first, Petakit 5D enables the real-time process and feedback for the microscopes, and also enables large-scale processing for petabyte-scale data. And these features are based on some innovative algorithms, including fast image reading and writing for TIFF and ZAR, combined geometric transformation, instantly converted deconvolution, and scalable stitching. And we also have a graphic user interface, so for the software for non-programmers without writing any code to run the software. And also we have a set of other useful tools for the basic image processing. So now let's go through these features to see how we can achieve this very amazing performance with an automatically faster. So first, we develop a generic computing framework to enable the processing of very large data using the whole computing cluster with multiple computing nodes. So in this framework, we have two types of jobs, conductor and work jobs. So first, the conductor job will accept the set of tasks, and then it will assign the task to the worker jobs, and each worker jobs will work on one task. After job submission, the conductor job will monitor the worker status, and then resubmit jobs if some fails, and then it will keep monitor the whole process until all the tasks are finished. In this way, the tasks are run in a parallel fashion using multiple nodes in the computing cluster. And this framework is very generic and can accept different types of tasks, like the separate images, like a time series, or small regions in a bigger image. So all our frameworks for the large-scale processing are built upon this generic computing framework. So now let's go through some of the algorithms that we have been developed in the software. So the first thing we have done is to develop fast image readers and writers for TIFF and ZAR. So TIFF is the traditional imaging format, and ZAR is the next generation imaging format that with better support for parallel image read and write, especially for large images. ZAR stores the entire images as small, non-overlapping regions, and each region in a small file within a folder. And this work was mainly done by my mentee, Matthew Mueller. He developed customized C++ code for the TIFF and ZAR readers and writers. So, and he also developed a Fiji plugin based on these readers and writers. So now let's first compare our plugin with the standard Fiji opener to open the same image. Let's see how long will it take. Yeah, our plugin only needs 4 seconds to open this image. And then let's see how long will it take for the standard Fiji opener. Yeah, seems pretty negative, right? Yeah, it takes 1.5 minutes. So this means this is at least 20 times slower than our plugin. And here's the more detailed comparison of the our readers with the conventional method. So let's set the relative performance for the standard TIFF reader libTIFF that you may have used even you don't know this library. Set the relative performance at 1 and the relative performance of our reader in C++ TIFF as green. And for all these different frames, our readers are over 20 times faster. And we do the same comparison for the TIFF writer, and our writer is about 7 times faster, and our ZAR reader is about 10 to 20 times faster, and our ZAR writer is about 5 to 8 times faster. So with the readers and writers, then we can go to the actual processing. So when we image a very large sample, due to the limited field of view of a microscope, usually we need to take a region by region with sample overlap, and then after imaging, we will put them together. So this is the stitching process. And so here's the basic scheme of our stitching pipeline. So first, we will convert the images from TIFF to ZAR to enable better parallel computing. After this, we will do some registration to refine the correlates of the tiles, because their correlates from the microscope are usually not 100% accurate. And after this refinement, we can stitch them together. And we do so by split the stitching task into many small tasks, with each worker work on one task. So all these 3 steps are like highly parallelizable, and we can use multiple nodes in your computing cluster to do the processing. And here's a comparison of our stitcher with the current method, big stitcher. And our stitcher is an order of magnitude faster. And after we post and submit our paper, so there were substantial updates in big stitcher. However, our stitcher is still about 2 to 3 times faster with their even than their newest version. And after stitching, the next step is deconvolution. So deconvolution is usually necessary for lattice lightsheet microscope images to deblur and reconstruct the real singulars. So the Richard-Knudsen deconvolution is the traditional method for the deconvolution. And this is our iterative method that usually requires tens to hundreds of iterations to converge. And this is a very slow process, even with CPUs. To accelerate this process, we develop a new deconvolution method by modifying a component in the traditional Richard-Knudsen method with the weighted winner filter. And it turns out that our method only needs 2 iterations to converge. So this is at least an order of magnitude faster. And here's the benchmark, for the comparison. And after deconvolution, the next step is skew and rotation. So for the next microscope, usually the light sheet have an angle to the curve slip, and these frames usually they are not in the same XY plane along the Z direction. But when we store the images, usually we directly stack these frames above each other along the Z-axis like this. So this means the image is stored in a non-standard geometric space, which will make the image squashed. So we need to do a transform to shift these slices to the right positions. So this is the deskew process. And after this, usually we will do a rotation to make the curve slip horizontal. And traditionally, these 2 steps are usually do separately. And we found this is very slow and memory inefficient, especially for large images with many Z slices, because it will generate a lot of empty space in the deskewed image. And actually, we figure out that we can combine these 2 steps together because both belong to the geometric transformation. But we need to do it smartly. And we found that when the step size is small, we can directly combine these 2 steps with no problem. But when the scan step size is large, if we directly do it, then there will be very obvious artifacts due to the interpolation errors. So we figured out that we can first interpolate in the raw data to make it as if the image is taken with a small step size and then do the combined processing. It turns out that both combined methods are much faster and much more memory efficient. So there's a benchmark, and for both combined methods, it's an order of magnitude faster, and it's become more and more faster when the number of frames increase and also become more and more memory efficient. So the methods I just described are most for the relatively small images that the processing can fit to the memory. For very large images, like terabytes to petabytes, that cannot fit to the memory, we have additional strategies. So for the stitching, the method just mentioned can already stitch very large data if the image tiles are small enough. And for very large tiles that the processing cannot fit in memory, we have a strategy that we will first downsample the tiles to generate some MIP snaps for the registration and distance transform to generate the weights for the fusion. And then we can use this information to stitch the original tiles. And for the deconvolution and discretization, we have similar strategies that we will split the data into small regions with maybe with some border buffers, and then with each worker work on a small region, and then we can merge them together in the end. Because we are using ZAR in this process, so this split processing and merge process can be done automatically. And we have benchmarked processing from 1 terabyte to 1 petabyte. It turns out all these processes are almost linearly scalable. So now we have gone through the major functionalities in the softwares. Now let me showcase some examples for the processing of large-scale data with the software. The first example is the large field view live imaging of peak amphitheater cells. So this dataset contains 301 time points with the total data size of 8.1 terabytes. And we use Packet 5D to process data. And then to enable real-time visualization, we collaborate with NVIDIA and use their software NVIDIA Index for the real-time visualization using multiple GPUs. And this allows to interactively visualize different regions in 3D at the full resolution. And we can see a lot of cell divisions, and we can also find some interesting events. Like we do find one that are cell divided into 3 doctor cells. And here we can also explore their dynamics at different angles with this software. So another example is imaging and the processing of the entire fly ventral nerve cord. So this data set is about 10 terabytes, and we use Packet 5D to process this data. And here we label a subset of motor neurons. And here we can see the bundles of axons. And we can also trace the axons at a long distance. And actually, this image contains several thousands of image tiles. After process, we can hardly see the gaps or borders between different tiles. So our software is also very generic and can be used to process other image modalities. So here is an example of the processing of the 2D face images. So after stitching, we can hardly see the borders between different tiles. And this also allows to inspect a lot of different events. Like we found an interesting thing that 2 cells go together and merge together, and also we can see a lot of cell divisions. So here's another example for the processing of the imaging of the mouse brain with 2-photon microscope. So this is also a large field view and with many tiles, and then we stitch them together. After stitching, we can hardly see their gaps. And this allows us to visualize the fine details in the mouse brain, like axons, dendrites, and dendritic spines. And then we can also explore and also trace the axons. So also, other than these major functionalities, we also develop some real-time feedback and processing for the microscope acquisition, because the mosaic can generate the imaging very fast, and also the raw data in the skill space. It's hard for the microscope operator to visualize and inspect the images during the acquisition. To solve this problem, we develop a real-time processing framework that we stream the data to the data server and using the computing cluster to process data during the acquisition. And then we can visualize the data and then provide instant feedback for the acquisition, like detect errors during the acquisition and correct these errors immediately. Or we can stitch different layers of the thin layers for the large samples to determine when the imaging is finished. So this is go deep to the layers until the end of the sample. So to summarize, we have developed a scalable and efficient software package 5D to process petabyte scale microscope images. It contains the most commonly used processing tools, including discrete rotation, stitching, and deconvolution. And also it contains some other useful tools like max projection, resampling, cropping. All of this are with support of large-scale processing. And this software can also be used for processing for other imaging modalities. And so here is the source code of our software in GitHub. And we also have user-friendly graphic user interface for long programs without writing any code to run the software. And we also have some Python wrappers for the important functions for Python users. So the manuscript of this work has been posted on bioRxiv. You are free to download if you are interested. So now we have the introduced our mosaic and also the package 5D tools, which represent the state-of-art Lattice Lightsheet and also the state-of-art processing tools. Now let's show how the combination of these two tools to the microscope and the software that enables the study of the mouse brain, more precisely the mouse effect bulb in unprecedentedly large scale and high resolution. That's the conventional microscope and the processing tools can hardly achieve. So first, let me introduce a bit of background about the expansion microscope, which was invented by Ed Boyden's lab in 2015. So in this technique, they will first mount the target molecules in an expandable gel, and after that, they will do tissue clear and gel expansion. In this way, the physical distance between the target molecules will be increased, make them more resolvable. So this is a physical way for resolution boosting. In 2019, Basic Lab and Boyden's lab collaborated to combine Lattice Lightsheet and expansion microscope to enable the super resolution imaging of large samples. So in their paper is an example of the imaging of the synaptic profiles along with axons in the mouse brain. This demonstrates that this technique can achieve synaptic resolution. However, even in this paper, their samples after expansion, the samples are still very small, most at a millimeter scale. This is most due to the limited working distance in the microscope. To enable imaging for even larger sample at like a centimeter scale, our collaborator Dr. Regal from the University of Illinois Chicago developed a new gel technique that this gel can be photocouplable with a laser, that we can cut gel with into pieces with a laser. And by combining mosaic with this new gel technique, it enables the continuous imaging of centimeter scale samples. So without resort to serial sectioning that may cause material loss or gel deformation. So here is an illustration of the imaging process. After expansion, we will use the mosaic to imaging the top layers of the sample by scanning through different regions. After that, we will use a laser to degrade the top layers of the image regions. This allows us to image deeper regions. And we will repeat this process, the imaging and the degradation, to ensure the imaging of the entire sample. And we have used this technique to image the entire mouse effect bulbs for both wild type and a mutated sample for the neurodegeneration disease model. And for each sample, it generates, we image exons and myelins. For each sample, it generates about half petabyte scale data with about 20,000 image tiles. And we use PetKit 5G to process the data set. And then we visualize the final products here. It's this generates can generate the unprecedented large scale and high resolution view for the olfactory bulbs. And here we also compare the performance of PetKit 5D with partnership methods. And in all these critical steps, our methods is an order of magnitude faster, and we require about 18 times fewer computing hours for the processing of one data set. And if we use some cloud computing for the processing, then we only need $4,000 to $6,000 instead of $6, $7, or $100,000 using AWS or Google Cloud. This means the traditional methods may burn out our money very quick if we have multiple samples or try multiple rounds for the processing. And but our methods can save a lot of money, and this makes large-scale imaging and processing more practical for the regular research labs. And if we want to do real-time processing, then we only need 10 line computing nodes instead of hundreds of nodes for the processing. And to understand the difference between the wild type and a mutated sample, we first do some macro scale analysis. So typically, the olfactory bulb can be classified as these several layers based on their features. So here's the list of the layers with their detailed name and also the main features. So to see how these layers change during neuron degeneration, we first manually annotate these layers, and we want to compare. We then calculate the percentage of these different layers within their own sample, and then compare the this volume percentages between the wild type and the mutated sample. Here we normalize the volume percentage against the wild type and we can see that in the mutated sample, the GL layer shrink a lot, but the GC layer increase a lot. So we will do further analysis to explore why this happens. And other than the macro scale visualization analysis, we can also crop some small regions and visualize detailed exon and myeline profiles between wild type and mutated type. So here we show the exon collaterals for the same layers or the exon projections between different layers. And we visually we can see some difference between these two layers, these two different samples. And we also segment and reconstruct the exon and myeline profiles and compare them. So first, we quantify the total exon length for the wild type samples and also for different layers. Also, whether they are myelinated that have myelin associated with the exon or not. And we can see the different layers, they may have quite different varying exon lengths. And then we also do some myelination analysis, compare the wild type and mutated sample for the different layers. And we can see compared to the wild type, the mutated sample have a significantly lower myelination ratio in the IPL and the GCL layer. This may suggest some association of the myelination ratio change during the neurodegeneration. We will further explore this in the future. So also this work has been posted on bioRxiv. You may read it if you're interested. So yeah, I would like to thank my advisors group and Eric for their generous support of the work for my research. Without their support, this work can never happen. And also I would like to thank the group members, both current and former ones, especially my mentee, Matthew Mueller. He did a great job for the software development. I would also like to thank the funding agencies for their support of the work. Yeah, so thanks very much for your attention for my work. I'm free to take any questions. Yeah.
Thank you very much. That was really fascinating presentation with a lot of really amazing tools that you've developed.
Yeah, thank you.
So the first is more of a comment, but I don't know if you want to add, I don't know if you want to ask this, Alex, or if you'd like me to read it out. I could.
I guess you're talking about me.
Yes.
Alex is a common first name, so I wasn't sure. Oh, I just yeah, it is only a comment. It's not a question. I observed towards the end of your presentation, you had a workflow where the top of a basically jelly block is imaged because you can penetrate into the sample with a certain depth. And then you use a laser to, I think you said, modify the top layer. I don't know if you're cutting it away, like ablating it, but anyway, I just found that technique interesting. I'm not used to seeing that with optical methods. There's something rather similar in a few EM methodologies, so it's creative to see this.
Yeah, actually, the animation is a bit misleading. Actually, we do the cut, yeah, like shown here. So we directly use a laser, just cut a thin layer and then remove the top image regions. So we do not do all the the degradation for the entire.
Oh, I see. Okay, so it's like used as a scalpel, but you still have to use a, I don't know, some needle or something to push away the
Oh no,
removed layer.
we just use the buffer to slash.
Oh, okay. It floats away. I see.
Yeah.
No, that's very interesting. It's a good idea.
We have another question from Jackie Ko. Did you want to unmute and ask your question?
Thank you for your work. I see that it's a kind of a called feedback microscopy. Basically, we sometimes call it smart microscopy. That we kind of do simultaneous descaling of the data and kind of detecting the event happening, interesting biological event happening under the microscope, and kind of focus on smaller regions to reduce the gigantic data generated out from the light sheet. But I'm curious if we kind of this feedback microscopy system you are saying is streaming the data directly to the cluster, and we also do have kind of infrastructure in our institute, but we are having a cluster with a dynamic resource allocation system. We most commonly use is using a Slurm system. I'm curious if your system is compatible with such kind of setting, or your system would require a dedicated computational node to do such kind of computation.
I think definitely it should be doable because actually our framework is also have the support of the Slurm. Yeah. And it's written in MATLAB, and we also have our like a deployed version with all need of the MATLAB license, and we also have a Python wrapper for that. Yeah, it should be doable, as I would say. Yeah.
Yeah, that would be helpful.
And yeah, also we optimize our tools very much. So usually a lot of steps are very memory efficient. If your image is small enough, then yeah, it don't need a very fancy hardware. Yeah.
Thank you.
Any other questions?
I have, looks like we have one from Matthias.
Matthias?
Yes, thank you very much. Great presentation, and it looks very exciting to have these kind of tools to help processing such big data, right? I mean, during your presentation, you only show processing data acquired with your Mosaic microscope, or maybe even when you had the collaboration with Edward Boyden, maybe the Lattice Lightsheet initially designed by Boyden. I was wondering if this tool, the Petakite 5D, would work also on other type of data, because it seems very powerful to be able to do this processing on clusters of computer, make it very efficient and saving a lot of time. So do you think, do you know if it would work on other type of file other than ZAR or TIFF?
Yeah, so actually works with other image models as we demonstrate here, like a face or 2-photon image data, right? And also in our paper, we have demonstrated with our like a data set from Allen Institute. This is about 100 terabytes, and yeah, we process this data with our tool. Yeah, so this actually this data set.
Oh, I see. It's because right now,
Yeah, so
as
this
the
data set 3 is from Allen Institute actually.
yeah, because so I was saying, as the Lattice Lightsheet technology is becoming a bit more common now, I mean, some manufacturers are already selling some, and so they have their own type of files. For example, in our institute, we have the one from Carl Zeiss. So I was wondering if, for example, files that comes from this microscope that are also Lattice Lightsheet, but not exactly the same modalities as the Lattice Lightsheet from the Mosaic or from the one from Beitzi's lab. Would it work also, or would we need to like modify a bit the code to process such data?
I'm not sure now because I have never worked with this commercial systems.
Okay,
I
sure.
think if they save as TIFF or ZAR, I think it should be work.
Okay, I
Yeah.
see. And do you know if it would work also with OME TIFF? I think it's like just some
Yeah,
type
it
of
works.
TIFF.
Yeah, OME TIFF is just a special kind of TIFF
Yes, but
with
it's just
some like metadata, additional metadata.
Yeah.
In our pipeline, we don't need to like read this metadata. We directly need to read the data, the actual image data. So yeah, it works. Yeah.
Oh, I see. Oh, so you don't use the metadata at all? You just use like directly the just the
Yeah.
images, like
Yeah,
the
exactly.
oh,
Yeah, yeah,
oh,
yeah.
I
But
see.
we have some parameters, so the user need to like
Yes, I was looking at your at the code last week, and I saw, for example, for the skewing,
Yeah.
you enter the angle at which
Yeah.
so I mean, if you compute the angles manually, then maybe you don't even need like the metadata or whatever. I mean, you can also like look into the resolution and the system.
Yeah. Peak size.
Oh,
Yeah, exactly. Yeah.
oh, I see.
Yeah.
I see.
Yeah.
Okay. Oh, thank you very much.
Yeah. Thanks.
Brandon, did you want to ask your question?
Yeah, I just had one question. On your new deconvolution method, the OWM or OMW, I suppose, is that also in the paper? I just didn't get to that part.
Yes.
Can you
Yeah.
tell me a little bit more about? I'm very interested in this.
Yeah. It's all these tools are in our paper. Yeah.
Okay. And so the biggest difference looked like you were masking in Fourier space. Is that what you were trying to do?
Yeah. So we first like we using the OTF to generate our to segment the OTF to get the support, right? Then generate some weights, and then we apply this weight to the winner filter, and then to generate this a back projector. Yeah.
Got it. Okay, interesting. Very cool. And then do you see the sort of the same types of artifacts that you would see? I suppose not, since it's no longer iterative.
It's very iterative, but only 2 iterations. In practice, actually, we see fewer artifacts in the native IMS, because if you do many iterations, the artifacts will be amplified a lot, right?
Yeah.
Yeah.
Okay. Awesome. That's really super fascinating talk. So thank you so much.
Thanks. Yeah.
Maybe if we have time for a quick answer to the last question from Alex Hall. Have you done any correlative imaging experiments such as X-ray, CT before doing expansion and optical microscopy?
No, we didn't do that because we are mostly focused on the like small biologic samples with optical imaging, right? We didn't do CT or X-ray for the that.
Okay. Well, thank you very much again for the presentation. We'll have the recording up on the BINA site soon. And I don't have a handy, what's the date of our next meeting, Brandon, do you know?
That I don't
No.
remember. I think it should be.
Everyone's on our mailing list, so we'll send it out in 2 months.
Well, it should be November.
Yeah.
Should be our next, probably the 10th, or around the 10th. We'll get it out to you. Sorry.
Yeah, sorry about that. All right, thanks everybody, and thanks again for a great presentation, Jiao Tao.
Yeah, thank you very much for the invitation. Yeah.